For over a century, archaeologists sifted through dirt, searching for bones and tools, confident that empty soil meant empty history. But new science has exposed a nightmare. Cave floors act as molecular sponges, storing human DNA for tens of thousands of years with no bones in sight. That means entire chapters of our story were scraped away unnoticed, buried in the dirt we have already thrown out. What secrets have we erased? And what happens when the only evidence left is invisible to the naked eye? Dirt is not just dead matter. In the right conditions, it acts more like a vault. Deep inside a cave, every grain of clay or fleck of iron oxide becomes a microscopic trap, ready to catch and hold fragments of DNA. These minerals, shaped over millions of years, have a natural knack for binding to the delicate strands of genetic material. Clay minerals, for example, use their charged surfaces to pull in DNA, locking it away inside tiny pockets and layers. The result is a kind of shield, one that blocks out heat, enzymes, and hungry microbes that would otherwise destroy genetic evidence in days. Goethite, a rusty iron mineral, forms tight bonds with the DNA backbone, making the molecule almost impossible to pry loose. Even calcite, the mineral that builds stalactites and cave walls, can wrap DNA in a crust of stone, preserving it for millennia. Scientists have measured these effects in the lab, watching as more than 90% of DNA latches onto clay at just the right pH. In real cave sediments, the combination of clay, iron, and calcium means that DNA can last far longer than anyone thought possible. The dirt itself becomes a molecular sponge, soaking up the genetic signatures of every creature that passed through. This invisible archive lies hidden in plain sight, waiting for someone to ask the right question, and to look not for bones, but for the ghosts they left behind. In 2012, a team of geneticists arrived at Denisova Cave in Siberia with a plan that defied a century of archaeological thinking. Instead of searching for bones or tools, they collected hundreds of sediment samples from the cave's ancient layers, some of them untouched by human hands for hundreds of thousands of years. These samples, no bigger than a handful of dust, held the potential to rewrite the history of human presence in this remote corner of the world. Back in the lab, the team began a painstaking process. Each sample was catalogued, dated, and prepared for DNA extraction. The results stunned everyone. In layers that had yielded no fossils, the dirt itself revealed the unmistakable genetic traces of 3 different human species. Denisovan DNA appeared first, dating back nearly 250,000 years. In higher layers, Neanderthal DNA surfaced, showing they too called the cave home around 200,000 years ago. Even more astonishing, traces of modern human DNA emerged in the youngest sediments. Altogether, more than 700 samples from 3 chambers were tested, and 175 showed clear evidence of ancient human DNA. Each one was a silent witness to a vanished population. For the first time, the cave's empty layers spoke. The dirt told of ghost populations, groups who left no bones, no tools, no carvings, yet whose genetic signatures still lingered in the mineral vaults beneath the surface. The evidence was undeniable. The absence of fossils did not mean the absence of people. In the hands of these geneticists, the cave floor became a record of lost worlds, its secrets drawn out grain by grain, molecule by molecule. Inside the genetics lab, the work begins long before a single test tube is opened. Every surface is wiped with bleach, ultraviolet lamps hum overhead, and the air itself is filtered to keep out stray skin cells. The reason is simple. The DNA trapped in ancient dirt is measured in femtograms, so little that a single modern skin flake, invisible to the naked eye, can drown out the genetic signal of a vanished population. Scientists dress head to toe in Tyvek suits, double gloves, and face masks, moving with the caution of forensic investigators at a crime scene. The extraction process relies on a chemical trick. Researchers use biotennylated probes, tiny DNA fragments designed to latch onto the genetic code of interest. These probes act like fishing hooks dropped into a slurry of dissolved sediment. If the right sequence is present, the probe binds to its ancient target allowing scientists to reel in fragments that have clung to clay or iron for tens of thousands of years. Even then, the risk of contamination never disappears. Any error, one breath, one careless touch, can ruin a week's work. To separate ancient from modern, the lab turns to damage signatures. Ancient DNA is battered by time, showing a pattern of cytosine to thymine changes at the ends of molecules. Only fragments with a post-mortem damage score of 4 or higher are trusted. Everything else is filtered out. This level of scrutiny means that every positive result survives a gauntlet of checks, controls, and computational filters. The result is a record that stands up to the fiercest scrutiny, equipping scientists and skeptics to weigh the evidence on its own merits. In the Astillero Mountains of northern Mexico, a cave called Chiquihuite has thrown the field of archaeology into turmoil. Researchers sifted ancient silts and clays, not for bones or tools, but for the faintest chemical traces of human DNA. The results were staggering. Fragments of Homo sapiens DNA were locked in the sediment, dated to nearly 26,000 years ago, thousands of years before the accepted timeline for humans in the Americas. The evidence was not a skeleton or a spear point, but a handful of genetic reads, dozens of human DNA snippets among millions of sequences from bats, bears, and plants. The signal was clear enough to survive a battery of contamination checks and repeated tests in independent labs. Yet the controversy exploded almost instantly. Critics said the case rested on a chemical whisper. No bones, no teeth, not even a scrap of burnt wood. The stone flakes found nearby could be geofacts, chips broken by falling rocks rather than by human hands. Skeptics demanded more: a jawbone, a hearth, a tool showing unmistakable human wear. Some questioned whether modern DNA from field workers or lab air had crept in, muddying the results. Others pointed to the cave's unstable layers, disturbed by centuries of burrowing animals and dripping water, as evidence that DNA could have migrated downward from more recent occupations. The stakes soared beyond a single cave. If the dirt story held, textbooks would need rewriting. A single question remained: can the chemistry in a gram of dirt outweigh a century of field tradition? For some, the Chiquihuite findings opened a door to lost chapters of human history. For others, they threatened to unravel the very standards of proof that science relies on. In the layered earth of Denisova Cave, the stone tools never changed. Blade shapes, flake scars, even the raw materials stayed constant through centuries of occupation. Genetic evidence told a different story. Deep in the sediment, scientists found DNA from one group of Neanderthals. Higher up, that genetic signature vanished, replaced by the DNA of a new population. No new tools appeared, and there were no obvious markers of conquest or extinction. Only the molecules in the soil recorded the turnover, a quiet replacement that left the surface undisturbed. Archaeologists trained to read the language of artifacts found themselves confronting evidence that could not be seen or touched. The dirt, once considered silent, became a witness to an upheaval that left no trace in stone. In these layers, the past revealed itself not through what people made, but through who they were. For decades, archaeologists measured success by the weight of bones and the count of artifacts, never guessing that the real archive was slipping through their fingers with every shovelful of dirt. Spoil heaps grew outside caves, tons of sediment were sifted for treasures and dumped as waste. The nightmare is clear now. Generations of scientists were trained to discard the very material that held the deepest record of human presence. As the truth came into focus, a new generation of researchers called for change. Field manuals were rewritten. Excavation teams began saving every gram of soil, logging its exact location and sealing it for genetic analysis. Some sites are even being re-excavated, their old spoil heaps probed for lost DNA. One field archaeologist put it bluntly: "We spent a century cleaning the artifacts and throwing the history in the trash. The lesson is raw and irreversible. The dirt was the library, and we burned most of the books." Right now, every handful of unearthed soil carries the potential to rewrite what we know about human origins. As ancient DNA unlocks invisible histories, the true challenge becomes what we choose to preserve or to discard next. Sometimes the answers we seek are what we are most likely to overlook.